दिख रहा है देखो स्क्रीन और आवाज वॉइस में आ रही है? चलो ठीक है, ओके, थैंक यू। Okay, good afternoon, students. Now, today we'll be discussing two fat-soluble vitamins which have been left yesterday, vitamin D and vitamin E. Okay, yesterday we discussed about vitamin A and vitamin K. Okay, now starting with vitamin D. Now, vitamin D, it was discovered in 1922 by McCollum. Okay, and how it was discovered? He found during his study, actually, he found that cod liver oil, on heating and oxygenation, it lost its xerophthalmia curing capacity, but retained its capability to cure rickets. Okay, so in this study, he found that like on heating and oxygenation of cod liver oil, the xerophthalmia capacity was being lost, whereas the capacity to cure the rickets was retained. Now, from this observation, he explained that xerophthalmia and rickets curing property of cod liver oil were due to different compounds. Okay, and the compound which was responsible for curing rickets, that is anti-rickety factor, was then named as vitamin D. Now, among the compounds which have vitamin D activity, like vitamin D1, D2, D3, okay, so out of these forms, two forms, vitamin D2, or we call it as a ergocalciferol, and vitamin D3, we call it as cholecalciferol. They are the most common organic compounds having the vitamin D activity. Okay, then vitamin D2 or ergocalciferol, it is the form of vitamin D, okay, that we get from plants, and vitamin D3 we get from the animal sources. Okay, now both vitamin D2 and D3, they are being derived from steroid. Okay, and in this what happens, as you know, the steroid steroid, it has four chains in it. It is a 17 carbon compound having four chains in it, A, B, C, D. Okay, so what happens in these two vitamins, D2 and D3, there is an opening of B ring at position C9 and C10, okay, in its steroid nucleus, right? So it is both, they are steroids, but they, with their B ring being open between C9 and C10. Okay, so as I told you that cholecalciferol, it is in a vitamin D form that we obtain from animal food, that is vitamin D3, and ergocalciferol, it is the vitamin D2, which it is the form of vitamin that we obtain from plant sources. Okay, now both these two vitamins, D2 and D2 and D3, they are equally potent, right? Now, this ergocalciferol, and it differs from the cholecalciferol. It has a structural difference, that it has an extra methyl group that is attached to C24 carbon atom in its steroid structure. Okay, and it also has a double bond between C22 and C22 and C23. Okay, now as they are being derived from a steroid, so they are lipid soluble vitamins. Okay, so D3, D4, why we call them? Because they have been derived from a sterol. Now, these forms, both vitamin D3 and D4, they are relatively resistant to heat and oxidation and to acidic and alkaline mediums. So, this vitamin D, it does not easily get destroyed either by cooking or in the preserved foods. Okay. Now, coming to the sources. Okay. Now there can be vitamin D can be obtained from both plant sources as well as animal sources. Okay, but animal source from plant sources, they are being sparingly see it is being obtained sparingly from the plant sources. Okay, because like vegetable oils, they are enriched sources of vitamin D2, and mainly it is being obtained from the vitamin D is from the dietary sources, the other animal sources. Okay, like we have fish liver oil, milk products, egg yolk, so they are a very rich sources of vitamin D3 or the polycalciferol. Okay, now this polycalciferol and argocalciferol, okay, they are in animal and in the plant sources, they are present in a precursor forms. Okay, so they need to be first converted to see we call it to their provitamin forms. Okay, before we uh, um, take up this vitamins in our body. Okay, through food. So what happens, uh, argocalciferol, it is being synthesized in plants from the argosterol, whereas in animals, in animal sources, or you can say in animal food, it is being derived from 7-dehydrocholesterol. Okay, so this conversion of argosterol into argocalciferol or vitamin D2 in plants, in plant food, it requires the sunlight and also the conversion of 7-dehydrocholesterol to polycalciferol or vitamin D3 in plant food, in animal foods, also requires the sunlight. Okay, because it happens, this conversion happens in the presence of, when they are being, these precursors are being exposed to the UV rays and sunlight. So that is why we call this vitamin as sunshine vitamin. Okay, so apart from the dietary sources, the source of vitamin D in our body is our skin. Okay, so in our skin also the vitamin D can be synthesized. Okay, now it can be synthesized in humans, and how does it be synthesized? With the help of sunlight. Okay, so what happens, like in skin, like malphizian layer of epidermis in our skin, the cells of malphizian layer, they have a precursor in them, that is 7-dehydrocholesterol. Okay, and which is an intermediate of a minor pathway of cholesterol synthesis. You might be knowing about it. Okay, because as you have read the lipid cholesterol metabolism. So this 7-dehydrocholesterol, when it is being exposed to the UV rays in sunlight, mainly the wavelength from 230 to 350 nanometers, then what happens, this 7-dehydrocholesterol, it gets converted into polycalciferol. Okay, that is vitamin D3. So apart from dietary sources, a very major source of vitamin D in our body is our skin, because in our skin it can be synthesized by 7-dehydrocholesterol uh, uh, when it is being exposed to UV rays from sunlight. Okay, now in 1930, it was observed that prevalence of rickets was much more in the European countries having the foggy weather than in developing countries like India. Okay, despite of high prevalence of malnutrition. So what? So this study actually, it showed that the deficiency of the vitamin D deficiency could be caused by the dietary deficiency, but could be because of the inadequacy in diet, but the level of exposure to sunlight is was more important determinant in maintaining the vitamin D levels in our body. Okay, so uh, uh, dietary sources are important, like in maintaining the vitamin D levels in our body, but the exposure of sunlight is a more important determinant than dietary sources in maintaining the normal vitamin D levels in our body. Okay, now One is, we have seen that exposure to sunlight. Exposure to sunlight is one of the important determinant in maintaining the vitamin D levels or in our body. The second one is the pigmentation of skin also play a very major role in maintaining the production of vitamin D3 in skin. Okay, so it has been seen that dark skinned people, they need to have a longer hours of exposure to sunlight than the light skinned people. Okay, so pigmentation of skin is also important. Now this diagram, it is showing basically the conversion of 70 hydrocholesterol into cholecalciferol, that is D3 in skin. Okay, so what is this? Is 70 hydrocholesterol. So with the help of UV rays in skin, which is coming from the sunlight. Okay, so this 7 dehydrocholesterol through process of photolysis, it is first get converted into an intermediate, that is sterol. Okay, so after that, because it is because it is unstable intermediate, then it spontaneously gets converted into cholecalciferol. Okay, which is vitamin D3. Right, so from sunlight, when there is exposure of skin to the sunlight, 70 hydrocholesterol, which is present in the cells of malpighian layer of skin, okay, so it gets converted into cholecalciferol or vitamin D3. Okay, now always remember that excessive exposure of sunlight, it never causes the toxicity of this vitamin. Okay, because because there is excessive exposure, then there would be an excessive formation of cholecalciferol D3, but this cholecalciferol would not deposit in our would not store in our body, because this would be otherwise being destroyed by sunlight. Okay, so excessive exposure of sunlight doesn't lead to vitamin D toxicity. Okay, now we have seen that vitamin D3 or cholecalciferol, it has two sources in our body. Okay, so either it can be obtained through diet or it can be synthesized in skin. But this vitamin D3, it is in the precursor form. This cholecalciferol, it is a precursor form. Okay, so it needs to be activated before this vitamin D3 or cholecalciferol, it exerts its biological actions on its different target tissues. Okay, so it is in a precursor form, or because vitamin D, it behaves like a steroid hormones. Okay, so we always sometimes call vitamin D as a hormone. So this vitamin D3 or the cholecalciferol, it is a prohormone. So it need to be converted into its active form so that it can have its various biological actions on different target tissues. Okay, so for this what is the activation? It needs to be converted. This cholecalciferol, it needs to be converted into 125 dihydroxy cholecalciferol. Okay, so we call it also calcitriol, because this structure, it has 3 hydroxyl groups in it, so we call it also as calcitriol. Okay, so it needs to be, this cholecalciferol needed to be get activated into its in its active form, that is 125 dihydroxy cholecalciferol, to perform its biological actions on its various target tissues. Okay, now coming to its metabolism. Now, as we know, it's a lipid soluble vitamin, so it is being absorbed along with the lipids in the diet. Okay, and predominantly it gets absorbed from the proximal GIT, that is from the duodenum, and jejunum. From there it is being absorbed. Okay, now once it is being absorbed, this dietary D3, it is being vitamin D, either D2 or D3, it is being absorbed. It then enters into the circulation through chylomicrons, because it's lipid soluble, so it needs to be incorporated into the chylomicrons, okay, to get released into the circulation. Okay, now once it is in the circulation, now because it is lipid soluble, so when it is being in circulation, when chylomicrons are being acted upon by the lipoprotein lipase enzyme to get converted into chylomicron remnants, so what happens, some amount of fraction of vitamin D3 or the cholecalciferol, it gets stored in the adipose tissues as cholecalciferol itself. Okay, but major part of this vitamin D3, it through chylomicron remnants, it is transported to liver. Okay, now here you can see this vitamin D3, it is being coming from diet, as well as which is being synthesized from the skin. Now the vitamin D3 or cholecalciferol, which is being synthesized in the skin, now how it is being transported? Now see, am I audible? Hello, hi Rameshwar. हेलो हाँ रामेश्वर, मेरा दिख रहा है क्या स्लाइड और आवाज सुन रही है? देखो एक बार ऑफलाइन, एक बार देखो, शायद मेरे को उसमें दिखा रहा था कि चेक योर कनेक्शन, एक बार देखो। तेरी डिस्कनेक्टेड दे रही है क्या? कोई कनेक्शन है नहीं? Okay, sorry, students, I got disconnected. Okay, sorry for the interruption. Okay, I'll continue further. So I was telling you that vitamin. So I was telling you what is the source of this vitamin D3, okay, in liver. So this vitamin D3, it is coming from the diet, okay, through chylomicron remnants, and the other sources, the one cholecalciferol, which is synthesized from the skin. Now from skin, how it is being transported? It is transported being bound to the vitamin D binding proteins. Okay, so once this cholecalciferol or vitamin D3, it is being transported to liver. Now here the first hydroxylation occurs. Okay, and this hydroxylation, it occurs at C25 carbon atom. Okay, so this is once it goes, this cholecalciferol, it goes hydroxylation at C25, then 25 hydroxy cholecalciferol is formed. Okay, and this happens with the help of enzyme 25 hydroxylase, and which is a cytochrome P450 and NADPH dependent enzyme. Okay, now this is the first hydroxylation which occurs in liver at position 25. Okay, now after the formation of 25 hydroxy cholecalciferol, it from liver, it enters into circulation. Okay, and here in circulation, again, because it's lipid soluble, need to be transported with the helper protein carrier. So here it is being bound to vitamin D protein, binding protein. It after binding to vitamin D binding protein, it enters into the circulation, and from circulation it reaches kidney. Okay, now in kidney, what happens? The further hydroxylation takes place, which is at position C1. Okay, and in this process, in this reaction, what happens? 25 hydroxy cholecalciferol now will be converted into 125 dihydro cholecalciferol or calcitriol. Okay, and this occurs because of enzyme alpha 1-alpha hydroxylase. Okay, so in this way, the cholecalciferol in its precursor form through two hydroxylation reactions, one at liver, first at liver, and second one at kidney. Okay, it gets converted into active form, that is 125 dihydroxy cholecalciferol. Okay, it is the active and the most potent form of vitamin D. Okay, then from here, from kidney, it's again this form, it enters into the circulation, transported through vitamin D binding proteins to its target tissues. Now, what are the target tissues of vitamin D? They are intestine, renal tubules, and bone. Okay, so these are the target tissues. Now, this enzyme 1-alpha hydroxylase, it is also seen to be found in bone and placenta. Now, in placenta, the function of this enzyme 1-alpha hydroxylase is that it helps in the fetal calcium metabolism regulation. Okay, so I hope now you have understood how the precursor form of vitamin D, cholecalciferol, it gets converted into 125 dihydro cholecalciferol or calcitriol. Okay, now as per the need of the body, if the vitamin D is being required for its biological actions, it gets converted into 125 dihydroxy cholecalciferol. Means the second hydroxylation takes place at C1 atom. But this hydroxylation can also occur at C23, 24, and 26 position instead of C1. Okay, so now, but these products which are being formed, they are inactive and they get further metabolized to water soluble compounds like calcitrienoic acid and get excreted. Okay, now this is a diagram basically which is showing how the vitamin D3 polycalciferol gets converted into its active form 125 dihydroxy cholecalciferol. Okay, so here you can see 7-dehydrocholesterol which is present in the skin in skin in the cells of Malpighian layer of epidermis. This 7-dehydrocholesterol, with the help of UV rays and sunlight, in skin it gets converted into cholecalciferol. Here you can see here is the opening of there is an opening of this ring, B ring, at position C9 and C10. Okay, so this then this cholecalciferol is formed. Okay, now this cholecalciferol once formed in the skin with the help of 25 hydroxylase enzyme, which is cytochrome P450 and NADPH dependent, it converts cholecalciferol into 25 hydroxy cholecalciferol. Okay, so this is one hydroxyl group which is being present in the in the 70 hydrocholesterol, and here you can see at position 25 there is an addition of one hydroxyl group. Okay, so in this way 25 hydroxy cholecalciferol will be formed. Then further hydroxylation occurs. This 25 hydroxylase hydroxylation, it occurs in liver. Okay, then the second hydroxylation, it occurs at position C1 and it is with the help of enzyme 1-alpha hydroxylase and it occurs in kidney. Okay, so you should always remember the two hydroxylations, the where what carbon atoms are involved and in which organ they are hydroxylation is being happening. Okay, so and ultimately then with the help of 1-alpha hydroxylase enzyme 25 hydroxy cholecalciferol, it gets converted into 125 dihydroxy cholecalciferol. We call it as we also call it as calcitriol. Okay, now you can see from this diagram, because next we'll be discussing about the regulation. Okay, now how this conversion of vitamin D3 is vitamin D3 into 125 vitamin D3, it is being regulated. Okay, so from this diagram you can see there are three steps at which the which can regulate the production of 125 dihydroxy cholecalciferol. Okay, the first step would be the exposure of light. Okay, if there is adequate exposure of light, then 70 hydrocholesterol in the skin would be converted into cholecalciferol. Okay, then second regulatory step is the conversion of cholecalciferol into 25 hydroxy cholecalciferol. Okay, यहाँ पे now we can see that this 25 hydroxy cholecalciferol which is formed, it is being responsible for the feedback inhibition. So it is being formed in excess. It can also inhibit this enzyme 25 hydroxylase. Okay, so it has an inhibitory effect on 25 hydroxylase enzyme. So this is the second step of regulation. Then third step would be the conversion of 25 hydroxy cholecalciferol into 125 dihydroxy cholecalciferol, which is happening with the help of 1-alpha hydroxylase enzyme. Okay, here now this is the step, most important step, which is being tightly regulated. Okay, because it is being affected by many factors. The activity of this enzyme 1-alpha hydroxylase is being affected by many factors. Okay, so here you can see the first is calcitriol itself. There is a feedback inhibition from the calcitriol. Okay, then we have other factors, stimulatory factors like parathyroid hormone, hypocalcemia, hypophosphatemia. Actually they have a stimulatory effect on 1-alpha hydroxylase enzyme. Now the vitamin D, what is the, we will just study in our next slide. What is basically the function of vitamin D in our body? Okay, basically it helps in maintaining the maintaining the normal homeostasis of calcium metabolism in our body, and it doesn't do it alone. It does this action with along with the PTH, that is thyroid hormone. So parathyroid hormone is equally important for maintaining the calcium metabolism in our body. So what happens whenever there are low levels of calcium in our body? This PTH, its secretion is increased from the parathyroid hormones. Then it has a stimulatory effect on it is one of the effect of parathyroid hormone, that by activating this 1-alpha hydroxylase enzyme, it increases its activity, due to which there is a more formation of active form of vitamin D3. Okay, that is cholecalciferol. So similarly, hypocalcemia, if there is a hypocalcemia, it will stimulate 1-alpha hydroxylase enzyme, due to which there would be a more formation of 125 dihydroxycholecalciferol, and which in turn then this vitamin D3, it helps in maintaining the normal maintaining the calcium levels in normal range. Now I'll tell you how this will happen in the next slide. Okay, again, hypophosphatemia, it has a positive or stimulatory effect on 1-alpha hydroxylase. Okay, so this is the most important step, okay, in the regulation of conversion of vitamin D3 into 125 dihydroxy vitamin D3, or we can call it as 125 dihydroxy cholecalciferol or calcium triol. Okay, so this all we have just discussed, that it can be regulated at many levels. One is that the conversion of 7D hydrocholesterol to cholecalciferol, which is dependent on the exposure to sunlight. Then it can be the conversion can be controlled by the feedback inhibition by 25 hydroxycholecalciferol on 25 hydroxylase enzyme. Okay, then we have seen that hydroxylation at C1 position is more tightly regulated, and depending upon the need in the body, 25 hydroxycholecalciferol is being formed differentially. Okay, either it get hydroxylated at C1 if there is a need in the body for vitamin D3, or it get hydroxylated at position 24 if it is not being needed. Okay, then 1-alpha hydroxylase in kidney. Okay, this is a very important, it is the most important enzyme which is regulating the metabolism of vitamin D3. Okay, so we have already discussed hypocalcemia, hypophosphatemia, and PTH. Basically, they stimulate this enzyme 1-alpha hydroxylase. Now, there also are some hormones like estrogen and other hormones. They also seems to be seems to have some have effect in the regulation of vitamin D. Okay, now coming to the mechanism of action of calcitriol or the 125 dihydroxy vitamin D3. Okay, so now let us see what is the mechanism of action of this vitamin in its active form in its various target tissues. Okay, now as I have already told you that because it is being a derivative of steroid, so its action is just like the action of steroid hormones. Okay, so that is why sometimes we also call that vitamin D as a hormone. Okay, so how does it act? It acts by modulating the gene expression of vitamin D responsive genes, okay, thereby affecting the protein synthesis, or a protein synthesis means effect and what would be that effect? Basically, it is increasing the synthesis of the proteins which are being encoded, which are being coded by these vitamin D responsive genes. Okay, so once being lipophilic, as it is a lipophilic vitamin, so it diffuses easily through its plasma membrane. So how does this action occur? How does it occur, this action of gene modulation? So being lipophilic, it diffuses into the plasma cells of target cells, okay, plasma membrane of target cells. Then once they are inside the cytoplasm of their target cell, they encounter very their specific receptors, okay, which have a very high affinity for vitamin, for the calcitriol. So they encounter their specific receptors, okay, and they bind to it. Now these calcitriol bound receptors, now which are in the cytoplasm, now they form a heterodimer, okay, they form a heterodimer with a retinoic acid receptor. If you remember, we have studied yesterday that vitamin A, it also acts through the cytoplasmic receptors, okay, and for retinoic acid we have two receptors, right? One is the all trans, one receptor is for the all trans retinoic acid and one is for the 9-cis retinoic acid, okay. So what happens, this calcitriol bound receptor, it binds to the receptor, vitamin D receptor, okay, vitamin A receptor, to which the retinoic acid is bound, okay, and then they form a heterodimer. Now this heterodimer then enters into the nucleus, and in nucleus what happens, this it binds to the DNA and the vitamin D response elements, okay. So once they are being bound to those vitamin D response elements or the hormone response elements, then it modulates the transcriptional activity of the vitamin D responsive genes and thus increases the mRNA synthesis of specific proteins, okay. So this is the mechanism of action. Now from this diagram I just wanted to make you understand so how it is happening. Here you can see this is a target tissue cell, okay. So यहाँ पे this is a vitamin D binding protein. The vitamin D here you can see it is in the calcitriol form, right? 125 dihydroxy cholecalciferol or the calcitriol form, and it is being bound to the vitamin D binding protein. So through this binding proteins they have reached the target tissue. Now in this is the cell membrane of the target tissue cell, right? So through this cell membrane it has now diffused into the cytoplasm. Because of its lipophilic nature, it has diffused into the cytoplasm. Now here you can see these are the vitamin D receptor. It is the vitamin D receptor 125 cholecalciferol receptor, vitamin D receptor, and here this is the calcitriol. So it binds to its receptor. Here the receptor binding it occurs. Then you can see here this is the receptor of vitamin A. Okay, so this is RXR receptor of vitamin A, that is retinoic acid. Okay, so to this receptor also the vitamin A is attached. Okay, so now this calcitriol bound receptor will bound to this vitamin A receptor, and then they form a heterodimer. Okay, now this heterodimer will move into the nucleus, and here in nucleus here they bind to the vitamin D receptor, vitamin D responsive response elements, okay, and thus they initiate the or they modulate the transcription of some specific proteins. Okay, so this is how the vitamin D it acts on its target tissues. Okay, now we'll see what are the actions of vitamin D on its target tissue. First we'll see what is action in intestine. Now in intestine what it does, calcitriol, it promotes the absorption of calcium and phosphorus from intestine. Okay, so how it does, actually calcium it diffuses passively from the luminal surface. Okay, it goes as it goes like along with its concentration gradient and diffuses very easily through the luminous surface of the intestine. Okay, then what happens when it is inside the intestine? Now this calcium, it is not able to now being released into the bloodstream. Okay, why? Because it is an energy dependent process. So from intestinal cell to blood, either it goes into the blood through sodium potassium, through sodium calcium exchange, or it being pumped out through the calcium cal binding complex. Okay, so there are two mechanisms. Now, where does this calcitriol it acts? It basically de-represses the gene which is coding for the cal binding protein. So once there is a de-repression, then what will do? It will stimulate the synthesis of mRNA for cal binding protein. Okay, so once there is a excessive stimulation, there is a stimulation of synthesis of mRNA for cal binding protein, so there would be an increased availability of cal binding in intestinal cell, and finally which will then help in the calcium absorption. Okay, so due to which the calcium absorption increases. Okay, so basically it is in a way it is creating a kind of a concentration in the intestinal cell. Okay, so this way calcitriol, it promotes the absorption of calcium and phosphorus from intestine. Okay, so the effect of vitamin D is in intestine is that it enhances the calcium absorption and thus helps in maintaining the calcium levels in blood. Okay, now next is action on bone. Now, what is the action of calcitriol on bone? What it does, as we know that in bone, two processes they are going on. One is the bone mineralization, and second one is the bone resorption. Okay. Now, through now this mineralization, it occurs through the activity of osteoblasts, and bone resorption, it occurs, it is being mediated by osteoclasts. Okay, now what does calcitriol do? Vitamin do? It basically promotes the bone mineralization by coordinating the remodeling action of osteoclast and osteoblasts. Okay, so it has a predominant effect, although it is having effect on both osteoclast as well as the osteoblast, but its predominant effect is on the osteoblasts. Okay, so which help in the mineralization of bone. Now in osteoclast, what it does, it promotes the differentiation of osteoclast precursors from macrophages, and to osteoblast, what it does, vitamin D, it enhances the synthesis and secretion of alkaline phosphatase along with the other osteopondin, along with other proteins, that is osteopondin and osteocalcin. Okay, so the action of vitamin D on bone is that it enhances the synthesis and thus the secretion of alkaline phosphatase enzyme from the osteoblasts. Now what is the action of now this enzyme alkaline phosphatase? Now what it does, because of its increased activity of this enzyme alkaline phosphatase in organic matrix of bone, what it does, it increases the local concentration of phosphates. Okay, so once there is an increase in the concentration of phosphates in the matrix, in the bone matrix, so what happens, the calcium and phosphorus, they reach to a supersaturated state. Okay, now once the supersaturated state is reached, mineralization, it favors the mineralization of bone. Okay, and due to which supersaturated state there would be a deposition of calcium hydroxyapatites, okay, over the organic matrix, over the bone matrix, okay. And as you have already read that bone matrix, it is made up of triple stranded collagen molecules, okay. So in this way, vitamin D, it favors mineralization of bone, okay. Now action on renal tubules. What calcitriol, it enhances reabsorption of calcium and phosphorus from renal tubules, okay. Thus it conserves those minerals by decreasing their urinary excretion, okay. Now as opposed to calcitriol, PTH also, it has a direct role. Parathyroid hormone, it also has a direct action on the renal tubules, but it only conserves the calcium, okay. It enhances the reabsorption calcium, but it not of the phosphorus, ठीक है. तो ये दोनों डिफरेंस है इनके बीच में, पीटीएच के और विटामिन डी के at the level of renal tubule, okay. Now other actions, these were the main actions of vitamin D. Other actions, there are also other actions, like it has been seen many tissues other than these tissues, target tissues, many other tissues, they have the vitamin receptors in them, but their function is not clearly defined till now, okay. Now in some studies it has been seen that cancer cell lines, like in cancer cell lines, when calcitriol was given, it was found to promote differentiation and was inhibitory for cell proliferation, okay. So sometimes it is being seen that the vitamin D also, it has a inhibitory role in the cell proliferation. So sometimes vitamin D, due to this role, it can be effective in psoriasis, a disease which is a hyperproliferative disorder of epidermal cells, okay. Now apart from this, vitamin D also helps in immune regulation, and okay. So this is all about the biochemical functions of vitamin D. So this here in this diagram, I just tried to just summarize what are the different actions of vitamin D in our body, okay. One is that immune regulation. Second, its action on intestine. In intestine, what it does, it increases the cal binding protein in intestinal cells, due to which there is an increased calcium absorption, okay, and due to which there could be an increase in the plasma calcium levels, okay. And in bone, what it does, basically it helps in differentiation of the macrophages into osteoclasts, okay, and also increases the synthesis of proteins like osteocalcin, osteopondin, and alkaline phosphatase from osteoblasts, okay, which are in turn are responsible for bone mineralization, okay. And then vitamin D also is known to have a role in regulating the epidermal proliferation of skin, okay. Then now, how do we measure the serum vitamin D3 levels? Okay, now we wanted to know. So, from understanding of the metabolism, we can say that, like, if we want to measure the vitamin D3 levels in our body, then we should measure its active forms. That is 125 dihydroxy cholecalciferol. ठीक है? But in actually क्या है कि like in lab we are not measuring 125 dihydroxy cholecalciferol to in order to know the vitamin D status in a person. Instead, we are measuring the 25 hydroxy cholecalciferol. Why? Because the T half of the 125 dihydroxycholecalciferol is very less as compared to 25 hydroxycholecalciferol. Okay, so most of the diagnostic tests which are measuring vitamin D3 levels, they are measuring 25 hydroxy cholecalciferol. Okay, now there are so many methods by which the vitamin D3 levels can be measured in blood. Okay, so here I have given the normal reference range, and it is being measured by a methodology, CLIA. Okay, so this reference range, they may vary with the different methodologies. Okay, so I have just referred here the values of which is being done by the CLIA. It is one of the methodology, that is chemiluminescence immunoassay, through which the vitamin D can be measured in blood. Okay, so here the normal reference range is up to 30 nanogram per ml. Now, when we say that the vitamin D levels, they are between 20 to 29 nanogram per ml, we say that the level is insufficient. Okay, now deficient, when we say when level there is a deficiency, when the levels they come they go down to 11 to 20 nanogram per ml of blood. Okay, and severe deficiency, when we say it is less than 10 nanogram per ml. Okay, and toxic levels, because it's a fat soluble vitamin, so if being taken in a very higher amounts, and mostly the toxicity, I'll just we'll just study in our next in slides. So toxicity can also occur with vitamin D. Okay, so this toxicity is when the levels they reach more than 150 nanogram per ml. Okay, now we'll discuss the vitamin D deficiency. Okay, what are the different causes for vitamin D deficiency? Now, as this age advances, there is a requirement for the vitamin D, it increases. Okay, so in advanced age, we can see it is a risk for vitamin D deficiency. Okay, then it has been seen that obese people, they are more prone for having this deficiency, because as we know that vitamin D can be stored in adipose tissue. So it has been seen that in obese people, since the release of vitamin D for the utilization is not easy, so this stored vitamin D cannot be used up. Okay, so which can then uh, lead to a deficiency state of vitamin D. Okay, then other factors which are responsible for maintaining normal levels of vitamin D in blood are, okay, are, we can say, like if there are intestinal diseases, if there is some malabsorption, despite of having adequate intake of dietary vitamin D, there could be an impairment at the absorption level, so that can also lead to the vitamin D deficiency. Okay, so the intestinal disease leading to fat malabsorption can lead to vitamin deficiency. Then if there are hepatobiliary diseases, okay, you say there is some liver disease, so what happens in that case? There would be inadequate bile, okay, in that, and also then because of this inadequate bile, then there would be an impaired absorption. Okay, then there is one more reason why in the liver diseases there could be a deficiency, because in that case the activation of vitamin D will also be affected, because the first hydroxylation that occurs at C25, it is occurring in the liver. So if it is not there, so it can lead to the deficiency. Okay, then renal disorders, again, because in renal disorders there would be uh, there would be impaired alpha 1 hydroxylase activity. Okay, and again it will affect the activation of vitamin D. Okay, so this activation which is occurring, hydroxylation which is occurring in kidney, okay, would not take place. Okay, so it will impair the activation of vitamin D, so due to which also the deficiency can occur. Okay, then other some drugs like phenobarbital, phenytoin, oral contraceptives, alcohol, they also influence the vitamin D levels. Now dietary deficiency, it could be a cause of vitamin dietary deficiency could be a cause of vitamin D deficiency, but the level of exposure to the sunlight, that is a more important determinant in maintaining the normal levels of vitamin D in blood. Okay, now there are okay, so these all are the causes which due to which there could be a vitamin D deficiency in a person, right? Now what are the clinical features, how does it present? Okay, clinical features may see in children the vitamin deficiency, the presentation would be different, and in adults it would be different. Now when it is being deficiency is presented in children, we call it as rickets, and in adults when vitamin deficiency is being there, okay, so then we call it as a osteomalacia. Now we'll see what happens in rickets. Okay, now in rickets, because of the vitamin deficiency, there would be an insufficient mineralization of bones in the child. Okay, so because of which there could be a skeletal deformities. Okay, and skeletal deformities, they are more common in children, okay, than adults in vitamin D deficiency. Okay, so in this what happens, because of the insufficient mineralization, the bones, they become very soft. Okay, they become very soft, and especially the bones which are being exposed to pressure, like skull bones, rib cage bones, right, they become deformed. ठीक है, तो deformed, और ये कैसे present करता है, अगर suppose frontal bone it presents as frontal bone prominence, जिसको हम frontal bossing कहते हैं, ठीक है, the child may have frontal bossing because of the frontal bone prominence. Then because of the deformity in the rib cage, the rib cage, it expands and becomes barrel shaped. Okay, just the shape of a pigeon. So that is why we call the shape as a pigeon chest shape. Okay, the child will have a pigeon chest. Okay, now what happens next, the bones which are weight bearing, okay, because in the insufficient, or you can say ineffective mineralization in these bones because of the vitamin D deficiency. So these weight bearing bones, they bend. Okay, they bend like bow. So we can see the bowing of legs in these children, and they have a knocked knees. Okay, then other deformity which could be there in vitamin D deficiency in rickets is the enlargement of epiphyseal ends of ribs. ठीक है, जो ribs के जो epiphyseal ends हैं वो enlarge हो जाएंगे, और costochondral junctions जो हैं वो भी enlarge हो जाएंगी. ठीक है, and this enlargement, it gives an appearance of beads. Okay, and they appear to be the ends of the ribs, they appear to be as a rosary. ठीक है, जैसे एक माला की तरह होता है, तो we call it as a rickety rosary. ठीक है, then vitamin deficient child, in them there could be widening of wrist, there could be loss of hair, they may have pot belly. And why there is a pot belly, there is a protuberance of abdomen, because of the decreased tone of abdominal muscles because of hypocalcemia. Because in vitamin D deficiency, what happens? There would be inadequate absorption of calcium and phosphorus from intestine and as well as from the renal tubules. So because of which normal calcium levels, they are not being maintained. So there would be a state of hypocalcemia. And overall, if we say, there would be a overall growth stunting. Okay, so these are all the features of classical rickets. Now, how it can be cured? It can be cured if we can fortify the food with vitamin D, or we increase the intake of vitamin D in diet. Okay. Now, these pictures I just wanted to make you understand just the symptoms. See, here you can see bowing of legs. The legs, they have become bow shaped. Okay, and here you can see the ends of the ribs because of the enlargement of the epiphyseal ends of the ribs. Huh? So there is a swelling, and such kind of appearance we call it as a rickety rosary. ठीक है, जैसे माला की तरह ये नजर आ रहा है. ठीक है, so these children would be having these clinical features. Okay, now since we have read rickets, so we should also be knowing about different types of rickets. Okay, so different types of rickets may just say, like one is the hypophosphatemia rickets. Now, why it is occurring? It is because, as the name implies, it is because of the defect in the tubular reabsorption of phosphate. Okay, there is a defect in the reabsorption of phosphorus from the renal tubules. Okay, so in this what we do, we give vitamin D along with the supplementation we give along with the phosphates. Then there is one more clinical condition, that is vitamin D resistant rickets. इसमें क्या होता है? इसमें basically vitamin D, it is adequate in diet, ठीक है, and see, but it is being not utilized properly by the target tissues. Okay, hello, hello, hello. Okay, sorry for the interaction. Okay, we were just discussing about vitamin D resistant rickets. Okay, so here what happens, the levels of vitamin D, they are normal in the body, but the vitamin D is not being utilized. Okay, it's utilized by the target tissues, right? Why it is so? It is because of the mutation in vitamin D receptor G. Okay, so because of which the vitamin D cannot be utilized by its target tissues for its biological for its various biological functions. Okay, so in this case, in vitamin D resistant rickets, what if we give vitamin D supplementation to the child would not improve. In fact, in this we have to infuse calcium and phosphates to cure this condition. Okay, to treat this condition. Then there is one more term, that is renal rickets. Now, what happens in renal rickets? This occurs because of the severe renal disease. Because we know that if there is a renal disease, then the activation of vitamin D3 into calcitriol would not happen, and this will then lead to the deficiency of vitamin D. Okay, so we call it as a renal rickets, and in this we calcitriol is being given as a treatment. Okay, now we will be discussing about the osteomalacia. Now, how does vitamin D deficiency? It is clinically presented in adults. Okay. Now, in adults, basically, if they are having deficiency They will complain of churnalized bony aches and pains. Okay, then there would be a reduced bone density. Okay, because of the insufficient mineralization. And this, how it is being seen, it is on x-rays, it is being seen as cortical thinning and reduced opacities of bones. Okay, so basically there is a see, organic matrix uh, would be normal, but the mineralization that would be affected. Okay, due to which there would be a reduced bone density. Right now, because of the reduced bone density, uh, uh, there is an increased tendency for bone fractures. Okay, now there is a term pseudo fracture that we usually see in adults having vitamin D deficiency. Now these pseudo fractures, actually they are not the fractures, but it is a radiological feature that we see on the x-rays in a vitamin D deficient person, and it is being seen in the bones like scapula, pelvis, femoral femoral neck. So what is that? Basically these are the marks which are being seen on the soft part of these bones due to the arterial pulsation against them, because the bone density becomes so less that a pulsation, arterial pulsations, they become visible. They make a mark, so we call it as a pseudo fractures. Okay, so um, then so this will all be the feature that would be seen in osteomalacia, right, in adults in vitamin D deficiency. Now what could be the biochemical alterations in vitamin deficiency? So there would be decrease in serum calcium, there would be decrease in phosphate, but the decrease in serum calcium would not be it would not be so markedly low that it would cause tetany, because this would be decrease in serum calcium would be then compensated by the overactivity of parathyroid hormone. Okay, so parathyroid hormone, if there is a deficiency of vitamin D, then there would be hypocalcemia in blood. ठीक है, so hypocalcemia की वजह से parathyroid hormone, now it will be there would be compensatory hyperplasia in parathyroid hormone, due to which there is a increased secretion of parathyroid hormone from the parathyroid glands, okay, and which then tries to maintain the calcium levels in normal range. Okay, so in vitamin D deficiency, we see decrease in serum calcium levels, decrease in phosphate levels, then decrease in vitamin D levels, and which is being measured by the form 25 hydroxy by cholecalciferol or 25 hydroxyl vitamin D3. Okay, then there would be increase in parathyroid hormone, and this is, we call it, when it is consequent to vitamin D deficiency, we call it as a secondary hyperparathyroidism, okay, because it is secondary to the vitamin D deficiency. And then there would be increase in the serum alkaline phosphatase activity. Okay, because it is also in compensation, because in deficiency there would be an compensation from the bone, so that more and more mineralization can occur. So this alkaline phosphatase activity, it increases. Okay, so these are the biochemical alterations that we seen in vitamin deficiency: decrease in calcium, decrease in phosphate, decrease in vitamin D levels, then increase in parathyroid hormone, and increase in serum alkaline phosphatase activity. Okay, and okay, now we have discussed secondary parathyroid para uh, thyroidism. ठीक है, तो there is a primary parathyroidism also. ठीक है, तो वो क्या होता है primary parathyroidism? उसमें क्या होता है, there is a primary reason, like there is a there is a disease in the parathyroid gland only, ठीक है, जैसे parathyroid adenoma हो गया, ठीक है. So that this basically is responsible, then this adenoma is responsible for excessive secretion of or secretion of parathyroid hormone from the parathyroid glands, okay. So the primary causes in the parathyroid gland only, okay. But in vitamin D deficiency there is a secondary hyperparathyroidism, okay, because it is occurring in consequent to vitamin D deficiency, okay. So I hope this is clear to you, okay. Then there are two more terms, like one is the hypoparathyroidism, which is just opposite to the hyperparathyroidism, and there is a pseudo hypoparathyroidism. In pseudo hypoparathyroidism, what happens? See, there is a parathyroid hormone, but because of the resistance in the target tissues, the parathyroid hormone is not able to act. So we call it as a pseudo hypoparathyroidism. Okay, so you should be knowing a little about hyperparathyroidism, primary and secondary, then hypoparathyroidism, hypoparathyroidism and pseudo hyperparathyroidism, okay. So then is what is the recommended daily allowance of vitamin D? So it is for children it is 600 international units per day. For adults it is 400 international units per day. Okay. In pregnancy and lactation it needs to be more. It is 600 international units per day. In elderly men and women, you can see here, as compared to the requirement of adults, it is more 800 international units per day with calcium supplementation. And obese children also, they need to have a higher supplement of vitamin D, like up to 800 IU per day, international units per day, okay. And then if it has been used for the treatment of osteoporosis, okay, then it has to be 1000 international unit per day to 50,000 international units every week, okay, every week. Okay, now we have seen that the requirement of the subjects who are sparingly exposed to sunlight is more. Okay, now we have understood why they would be requiring more vitamin D as supplements if they are being sparingly exposed to sunlight. Okay, but the recommendation for them has not yet been defined clearly. Okay, now coming to vitamin D toxicity. Now how this toxicity occurs, I have already told you, it never occurs with the either with the dietary sources because of the increased intake of food, or it never occurs because of the excessive exposure of the skin to sunlight. Okay, it occurs only when we take higher doses of vitamin D as supplements for curing different diseases. Okay, so like we have read vitamin D resistant rickets, like in hypoparathyroidism, in renal osteodystrophy, in osteoporosis, in which basically we give vitamin D as in supplements, and when they have been given in a very high doses for a very long time, then it can cause toxicity. Okay, because being lipid soluble, then it gets stored in the body and can cause toxicity. And then apart from this, vitamin D toxicity can also occur when we take food which have been fortified with excessive vitamin D. Okay, and we take it then regularly. So even with the fortified food which has been excessive, which is having an excessive vitamin D in them, and we take them with them regularly, can lead to the vitamin D toxicity. Okay, so see, like in countries where the incidence of rickets was very high, so these countries, they like started giving the vitamin D fortified milk and other food to their population. But what happened, because in like it has been seen in some cases the vitamin D toxicization occurred. Okay, that because of the indiscriminate fortification of milk with excess of vitamin D. So what happened? Some countries then they stopped giving the food fortified with vitamin D. Like in Europe, the vitamin D fortified milk, it is being banned. Okay, for this reason, because it, if being given as an indiscriminate fortification, can lead to toxicity. Okay. Now, what would be the clinical features if the toxicity occurs? There would be nausea, vomiting, hypercalcemia, because if there would be more vitamin D, then the levels of calcium will, serum blood calcium levels will rise. Okay, because of which there will be hypercalciuria, then thirst, polyuria, muscular weakness, and there could also be occurrence of stones in kidney. So that is we call as nephrocalcinosis. And because of the hypercalcemia, there could also be the calcification of soft tissues. Okay, like heart and lung. Okay, and then in ECG also, because of hypocalcemia, there could be some changes that can occur because of the vitamin D toxicity. Okay. Now in toxicity, we have it has been seen that the blood levels of 25 hydroxycholecalciferol, they are 15 times more than the normal levels than 125 dihydroxycholecalciferol. Why? Because the T half of the 25 hydroxycholecalciferol is more than the T half of 125 dihydroxycholecalciferol. So it tries to build up 25 hydroxycholecalciferol. It tries to, if there is a toxicity, so it tries to build up in the tissues. Okay. So so whatever features like we see in the vitamin D toxicity, they may be the features, this 25 hydroxycholecalciferol, it might be mimicking the action of, it is because of the calcitriol mimicking action of 25 hydroxycalciferol. Okay, cholecalciferol. So basically the toxicity which is occurring, it is because of the calcitriol mimicking action of 25 hydroxycholecalciferol. Okay. Now treatment, what would the treatment? Withdrawal of vitamin D. Okay. Then reduction of dietary calcium, and if not treated, in severe cases, glucocorticoids and calcitonin are useful, because glucocorticoid, they antagonizes the action of vitamin D, and calcitonin, it is a hormone which reduces the blood calcium levels. Okay, so in severe cases, glucocorticoids or calcitonin would be, in severe cases, would be useful. Okay. Now with this, now I complete vitamin D. Okay. Now one vitamin is left. I have to complete that also in this class, or in this class only. So you have to be little patient. Okay, because I have already been taken 1 hour. So you just have to be a little alert to listen to one more vitamin which is being left. Okay, that is vitamin E. Okay, so now we will be discussing about vitamin E. Now vitamin E, this is also a lipid soluble vitamin. Okay, and it is the most powerful natural antioxidant. इसका जो main action हमारी body पे है, that is, and it acts like a antioxidant. ठीक है, it is a most powerful antioxidant. It has an antioxidant activity in our body. Okay, now first it is being isolated from wheat germ oil. ठीक है, and being given a name, tocopherol. Okay, a substance which has been isolated and been given tocopherol. Now, why the vitamin E are tocopherols? They are being called as vitamin E being called as tocopherol because toco is means childbirth, ferosis means to bear, OL is alcohol. Okay, so basically vitamin E, it is a anti-infertility vitamin. Okay, because in studies it has been shown that if vitamin E deficiency was induced in lab animals, it resulted in infertility. Okay, so from this, they it has been now given this name. Vitamin E has been given a name, anti-infertility vitamin. Okay, the vitamin, it is a group of compounds, refers to the group of compounds like tocopherols and trionols. Okay, basically these all are the compounds, tocopherols and tocotrienols. They have a vitamin E like activity. These they all these compounds, they they differ in their biological potencies or activities, and out of there are 8 tocopherols which have been present in nature, which possess the vitamin E activity, out of which most active vitamin E is the D alpha tocopherol. Okay, so most active which is naturally present, it is D alpha tocopherol. ठीक है, अब कुछ सिंथेटिक भी होते हैं tocopherols, they are which has been used therapeutically. Okay, so these, but synthetic tocopherols like DL alpha tocopherol, they do not have the similar biological potency as the natural tocopherols have. Okay. Now, what is the structure, chemical structure of vitamin E? As we know that it is a fat soluble vitamin. Okay. So it would be having an isoprenoid compound in it, right? So basically, this vitamin E, it is a isoprenoid substituted 6-hydroxychromates. Okay, means this is isoprenoid unit, and this is being attached to this 6-hydroxychromate. This cyclic structure is 6-hydroxychromate. You just have to remember just need to understand this. You don't have to remember. Okay, so these are basically isoprenoid vitamin E compounds. They are isoprenoid substituted 6-hydroxychromates. And as we have seen, out of all the tocopherols which exhibit vitamin E activity, D alpha tocopherol, it is the widest, it has the widest natural distribution and has the greatest biological activity. Okay. Now, metabolism of vitamin E, digestion, how digestion and absorption, it gets digested. Mostly the vitamin E, it is present in vegetable oils. Okay, it is in the plant sources. They are the rich sources of vitamin E present in vegetable oils. Okay, now what happens? How they get absorbed, digested and absorbed. They are in vegetables oil. They are being dissolved in dietary fats. Okay, as tocopherol esters. Then once there is an intake of this plant food, okay, of intake of vegetable oils, these they disperse vitamin E in the form of tocopherol esters. They disperse in bile in intestinal lumen along with the other dietary fats. Okay, then what happens? They get then liberated from the fat in the intestinal lumen, and then once liberated, they get directly absorbed through the intestinal get directly absorbed into the intestinal cell. Okay. Then what happens in the intestine? Inside the intestinal cell, they get incorporated, as happens with all other fat soluble vitamins. They get incorporated into chylomicrons, okay, because they are fat soluble, so need to be transported in blood, so they get incorporated in chylomicrons. then transported to bloodstream and get different to then get distributed to different tissues, okay, having the lipoprotein lipase. And then after the action of lipoprotein lipase on chylomicrons, the chylomicrons now remnants, they then being transported to liver. Now these chylomicron remnants would be having tocopherol in it, okay. So this tocopherol now will be taken up, remnants will be taken up by the liver, and by the liver, and inside the liver, what happens? They get incorporated into the VLDL, okay, then to get exported to the target tissues, right? So कैसे होता है vitamin E कैसे transport होता है to the target tissues? By incorporating into VLDL, ठीक है. इनके लिए it doesn't have a specific binding protein, but through VLDL it reaches its target tissues, okay. Now excess of vitamin E, it get stored in adipose tissue, okay. Now rest, we have just yesterday we have seen in, I have told you in the introduction part that vitamin A, D and K, they get stored in liver, whereas the major tissue which stores vitamin A is adipose tissue, okay. So whenever there is a need, this vitamin E will be then released from the adipose tissue, ठीक है. So transport and storage ये हम लोग ने अभी पढ़ ही लिया है, ठीक है. Now how does vitamin E deficiency occurs? Now this vitamin E deficiency, otherwise it seldom occurs, okay. Why? Because vitamin D, it has in our body, there are adequate stores of vitamin E in adipose tissue. Number two, vitamin E is widely present in, it has a very wider distribution in nature, okay. So dietary deficiency भी बहुत कम होती है vitamin E की, okay. So unless until there is some other condition which is impairing the absorption or the transport or the utilization of vitamin E, this vitamin E deficiency will not occur, okay. So what are the conditions which can lead to vitamin E deficiency? Again, impaired, where the condition in which there is impaired absorption of this vitamin, जैसे chronic steatorrhea हो गया, A beta lipoproteinemia हो गया. Because if there is A beta lipoproteinemia, then it will not be transported to its target tissue because it is being transported in VLDL, okay. Then cholestatic liver diseases हैं, or so all these diseases which are affecting the absorption and transport of the vitamin E can lead to vitamin E deficiency, okay. Now how it is get catabolized? It can catabolized by its oxidation. When its oxidation of bromine and of the side ring occurs, okay, first the bromine and side chain, it gets oxidized and then it conjugates with the glucuronic acid and get excreted in the urine, okay. Now most important, what is the biochemical role of vitamin E? As we have seen, vitamin E, it is a very powerful antioxidant. So basically vitamin E, it is the first line defense in our body against the lipid peroxidation of polyunsaturated fatty acids present in cellular or in subcellular membranes, okay. So basically it quenches the action of free radicals in the cells, okay. And it is vitamin E, they have a very high affinity to get concentrated in the cellular membranes or in the subcellular membranes because of its lipophilic nature, okay. और इसी में ही जो cell membranes हैं, as I think you might have read in the lipid metabolism, okay, how does the lipid peroxidation occurs? Okay, तो क्या होता है कि cells में there is a continuous generation of free radicals. ठीक है, free radicals. Now free radicals, they are very reactive compounds. Okay, so because they are being continuously generated in the cell, so they attack the polyunsaturated fatty acids which are present in the cellular or in the subcellular membranes. Okay, now if and because of which this attack of free radicals, they convert these PUFAs, they cause a peroxidation or the oxidation of these polyunsaturated fatty acids, okay, resulting into the formation of lipid peroxides. So these free radicals, they form the lipid peroxide. Now these lipid peroxides are also in turn are very reactive molecules. ठीक है, तो क्या होगा कि if these lipid peroxides, they are not being reduced or being acted upon by the antioxidants, so what will happen, this lipid peroxide with further being a reactive free radical will act on other polyunsaturated fatty acids which are present in the cell membrane or in the subcellular membranes. Okay, and will keep on, then this process will keep on continuing. It becomes a chain reaction. Okay, one PUFA is leading to one lipid peroxide is leading to the generation of another lipid peroxide, then this lipid peroxide another will be attacking other PUFA molecule in the membrane, and in this way a chain reaction will occur. Okay, so vitamin E, where does it occur? It is a chain breaking antioxidant. Okay, it is a chain breaking antioxidant. Basically it breaks the chain of lipid peroxidation. अगर lipid peroxidation होता जाएगा तो क्या होगा, there would be a damage to the phospholipid membrane phospholipids or the subcellular membrane phospholipids, and ultimately it can cause a damage to the cell. Okay, so vitamin E, it is acting as a chain breaking antioxidant, and it acts as an antioxidant in those cells which have a very high concentration of oxygen. Okay, so and it tends to protect the lipid structures in those cells which are being exposed to very high oxygen partial pressures, like RBCs, RBC membrane, retina, respiratory tree membranes. So these membranes, they are being exposed to very high oxygen partial pressures. So yeah, so this side, the vitamin E, it plays a major role of being acting as an antioxidant. Okay. Achha, this I just wanted to just show you how this peroxidation is taking place from a free radical. Here, this is a free radical. It is acting on this PUFA, polyunsaturated fatty acid, and converting it into a free radical. Okay, okay. Then this will be acted upon this by the oxygen. Okay, and will convert it into a lipid peroxide. So this is a lipid peroxide molecule of PUFA. Now this lipid peroxide molecule, then what will happen? This will then can propagate this reaction. This will act on other PUFA molecule and convert it into a free radical, and convert that PUFA molecule into another free radical. So in this way, the chain reaction will go on. ठीक है, तो विटामिन ई कहाँ पे एक्ट करता है? विटामिन ई एक्ट करता है इस स्टेज पे, एट द प्रोपगेशन स्टेज ऑफ लिपिड पेरोक्सिडेशन. ठीक है, और लेकिन कुछ जैसे एंटीऑक्सीडेंट्स हैं which are known as preventive antioxidants. ठीक है, वो preventive antioxidants है which prevent. So they act in this stage, in the initial stage of lipid peroxidation. Suppose like we have a catalase, some peroxidases, they are the preventive antioxidants. Okay. Now in this diagram we can see that this is a free radical, how it is quenching it. It is being see this unpaired electron, it is being taken up by the tocopherol, okay, and it gets reduced, and this now the tocopherol which is formed, it is a non-reactive uh, free radical product. Okay, it is less reactive than the PUFA radical or than the lipid peroxide radical. Okay, now vitamin E is actually, it is a aqueous, it is a lipid, it acts in the lipid phase. Okay, because it is in the membranes, it mainly acts in the membranes, in the cellular membranes, because there the environment is lipophilic. So vitamin E, you can see here, this is a cell membrane. Here the PUFA molecule, which is being acted upon by a free radical, and with the help of oxygen, this PUFA is converted into lipid peroxide. Okay, you convert ho gaya. Now this lipid peroxide now will be converted into lipid hydroperoxide, get reduced into lipid hydroperoxide, and this happens with the help of tocopherol. This tocopherol, it has given its one reducing equivalent, and it itself gets oxidized, right, and gets oxidized. Okay, now so that the function of tocopherol, it continues as an antioxidant. Now this oxidized tocopherol, now in the cell membrane, it needs to be regenerated back to its reduced form. Okay, so how does it occur? How does it occur? It occurs with the help of vitamin C. Okay, so vitamin C, it takes up a reducing equivalent from the reduced vitamin C, and it gets converted into reduced tocopherol. Okay, and the vitamin C itself, it gets oxidized. Okay, so it needs to be regenerated. तो कौन regenerate करता है इस oxidized tocopherol को? With the help of vitamin C. Then glutathione also, reduced glutathione also, it provides reducing equivalents to oxidized tocopherol, and it gets regenerated back to its reduced form. Okay, so you should remember this diagram, it's very important. Okay, so as basically there is an interplay of various antioxidants in the cell. Okay, due to which the different free radicals, they are being quenched. Okay, their action has been quenched. Otherwise, these radicals, they will cause damage to various cellular components like lipids, okay, like lipids, proteins, and DNA, and can ultimately lead to the cell damage. Okay. Then here, you can see, once this PUFA hydroperoxide is now formed, it will, by the action of lipoprotein lipase A2 enzyme, you might have read in lipid metabolism, it gets hydrolyzed or detached from the phospholipid, from the membrane phospholipid, okay, and then it enter into cytoplasm, and further then it is being acted upon by catalase and get converted into hydroxy PUFA, okay, and which is a non-free radical product. So in this way, a lipid peroxide radical is being converted into a, which is a very highly reactive free radical, is being converted into a non-free radical product. Okay. So apart from having antioxidant activity, vitamin E, it also reduces the risk of atherosclerosis, okay, by reducing the oxidation of LDL. How do, okay, because LDL also contains the has the phospholipids in it. Okay, so free radicals, they can also act on these can act on the PUFA which is uh, present in the phospholipids of these lipoproteins. Okay, so and atherosclerosis ka one of the major cause of development of atherosclerosis are the oxidized LDL. Okay, so basically this vitamin E also reduces the risk of atherosclerosis by reducing this oxidized LDL. Okay, a deficiency. Coming to the deficiency, as we have seen, no major disease has been found to be associated with the vitamin E deficiency in humans. Okay, because of the adequate diet, the adequate levels in the average diet, and they because of the adequate stores. But there are some clinical conditions with which deficiency occur that we have already discussed. Okay, so what could be the clinical manifestation? Poor nerve conduction. The patient may suffer from this spinocerebellar ataxia. They may have retinopathy, myopathies, or anemia. Okay. Now, what is the required dietary allowance? This is for males, it is 10 milligram per day, females 8 milligrams per day. During pregnancy, it is 10 milligram per day. During lactation, it is 12 milligram per day. Okay. Now, hypervitaminosis, again, it is a when it is being given in the clinical conditions where is deficiency only when given in a very higher doses for a prolonged time, then it can cause hypervitaminosis. Okay, when it is being given as a supplement, only the supplementations can cause the hypervitaminosis. And in this, if there is what happens in hypervitaminosis, there is a tendency for hemorrhage. Why? Because vitamin E also it has a mild anticoagulant activity. Okay, so with this I complete fat soluble vitamins. Okay, so if you have any doubt, please you can write here, or you can put your queries in the group. ठीक है, जो आप लोगों का ग्रुप है ना, you can also write their queries there. ठीक है, तो अभी if you have any query, you can ask me. Vijay, have you written? I could not see. Have you written your doubts? Yes, there is one question. If vitamin D tablets are being just a minute, given without any dosage, can be manually to I'm not getting this question, can be taken manually to, is it right? See, no, actually vitamin D, it has a specific dose. Okay, and it needs to be given in that dose only or for a particular time. Okay, it should not be taken uh, continuously, because being lipid soluble, it can be stored in the body and can cause toxicity. Okay, so there should not be indiscriminate use of these supplements. Okay, and it should always with in consultation with the doctor. Okay, right? Okay, bache, like if I'm not able to see respond to your queries now, you can put in the group. Okay, I try to solve, I try to discuss those queries with you in group. Okay, because I'm not able to see. Somebody has asked about 11 cis, and what is that question? Is 11 cis retinal and all transretinal interconvertible or unidirectional or unidirectional? 11 cis retinal and all transretinal, but they are interconvertible, and we need to have an enzyme for this is isomerase enzyme. Okay. How does it control? Uh, Could you repeat? Actually, some student has asked about uh, role of vitamin in radical PUFA. But see, radical PUFA is that basically they give their vitamin A, give its reducing equivalent, means one hydrogen atom which is having a what are basically the free radicals? Free radicals are the compounds which are having unpaired electron in it. Okay, so that property of this having an unpaired electron in it makes these radicals very reactive. Okay, so what this vitamin E does, it donates one hydrogen, okay, to it, and then quenches the makes that lipid peroxide uh, that the lipid peroxide radical converts into a lipid hydroperoxide. Okay, and this hydroperoxide, it is less reactive free radical than the lipid peroxide radical. And this process, during this process, it itself gets reduced, it gets oxidized, it gets converted into free radical, but this again get regenerated. This oxidized tocopherol, it gets regenerated back with the help of vitamin C. Okay, vitamin C and reduced glutathione, which is are the C antioxidants present in the cytoplasm. Okay. Any other doubt, bache? And if not, I'm not clarified every query, so you can write in the group. Okay, and then we can discuss there. Okay, one student is writing, do we need to remember all the synthesis and interconversion cycles in this chapter? बच्चे देखो इसमें important जो है fat soluble vitamin में we start with the vitamin A. You should be knowing the role of vitamin A in vision, in dim light. Okay, उसमें आपको पूरा पता होना चाहिए. Then Vanck's vicious cycle you should know. Okay, then in vitamin A deficiency manifestations you should know, and in vitamin D you need to know, yes, that is very important, how the vitamin D3 it gets converted into 125 dihydroxy vitamin D3 in its active form. That's very important, it may be asked. Okay, वो है. फिर mechanism of action, basically of mechanism of action of these fat soluble vitamins. Okay, so how do they all have a common mechanism of action, so you should be knowing about it कि कैसे होते हैं, how do they act. Receptors से जाता है, receptors then they go to the nucleus, then this bound vitamin D it bind to the HRE DNA, the HRE of DNA, okay, and then they modulate the gene expression specifically for the vitamin A and D. Okay, E and K में अगर आप देखते हैं, अच्छा D में ये हो गया, then you should be knowing about the clinical manifestations in vitamin D deficiency. Okay, वो आपको पता होना चाहिए. Vitamin K में you are talking about the cycle is the vitamin K cycle that we have read yesterday. कि how the hydroquinone, ठीक है, it gets regenerated back. This quinone means vitamin K. It gets converted back to the hydroquinone. Okay, the conversion of glutamic acid into the alpha glutamyl glutamic acid. That reaction is very important. You should know how does it occur post transitional carboxylation. How does it occur in the clotting factors? Okay, वो है आपको पता होना चाहिए. Then you should be knowing the mechanism of action of anticoagulants. कल हमने पढ़ा था कि how do they act. ठीक है. Then vitamin E में कि how do they, vitamin E it acts like a antioxidant. ठीक है. तो this is you should be knowing in vitamin D, in fat soluble vitamin D. Okay, बाकी then you can have your queries, you can send in the group. Okay, so thank you so much for with me and for last 2 days. Okay, you are with me and hope you have understood this topic, and for any queries always welcome. You can either come to me and ask, or you can now everything is online, you can ask on the group. Okay, so thank you so much, thank you so much for listening me patiently. Okay, so okay, thank you.